Good afternoon. Welcome to first virtual conference of ISV Global Vaccine Partnership. Over the last year, ISV established a committee on global collaboration and partnership. New Global Vaccine Partnership Program was established. Our mission is to build a global community of vaccinologists and vaccine supporters. This program will engage and partners with vaccine groups globally. We'll promote communication and collaboration among organizations who seek to innovate, develop, manufacture, and promote vaccines. This program will support development and the use of vaccines to prevent and control infectious and non-infectious diseases in animals and humans. Today's meeting, we are fortunate to have 8 organizations from different parts of the world to share their wonderful work in vaccines. This conference will be chaired by ISV Committee of Global Collaboration and Partnerships. You will find the name of chairs and participation groups in the next slides.
Hello, it's my pleasure to introduce today Dr. Jerome Kim from the International Vaccine Institute, IVI. Jerome is one of the most respected leaders in global vaccine community and currently serves as the Director General of IVI. He has made remarkable contributions in advancing vaccine research, production, and equitable access worldwide, particularly in the field of emerging infectious diseases and global health equity. His presentation today is titled Discover, Develop, Deliver, Empower, the global mission of the International Vaccine Institute. Please welcome Dr. Jerome Kim.
Good day. My name is Jerome Kim, and I'm the Director General of the International Vaccine Institute. I'd like to thank the ISV for the opportunity to speak today about IVI and the work that we do in support of vaccines for global health. IVI is a UNDP chartered international organization dedicated to accelerating vaccine research and development for global health. Our mission is to discover, develop, and deliver safe, effective, and affordable vaccines, empowering vaccine equity, impact, and sustainability globally. What you see on the figure below under discovery, development, delivery, and epidemiology and surveillance are the things that the 290 staff at IVI do on a day-to-day basis. Each of the blue boxes can be broken into separate modules to train people, other people, in the work around end-to-end vaccine discovery, development, and delivery. What you see in gray are the diseases that we're developing vaccines for. Under discovery, these are vaccines that are still in preclinical or in the laboratory. Under development are vaccines that are in human clinical trials. Under delivery, vaccines that are approved already, where IVI's work is supporting the generation of real-world evidence that may support either improved indications for the use of the vaccine or may change the way we use vaccines. So, for instance, human papillomavirus vaccine was tested with the Ministry of Public Health in Thailand in order to generate data around the use of a single dose of HPV vaccine, and the data were able to be used by the World Health Organization to help to change the recommendation from 2 doses to a single dose of HPV vaccine. Under epidemiology and surveillance, you see a list of diseases that is, I have to say, ever-changing. Why? Because the epidemiology group at IVI helps us to understand what the true burden of infectious diseases is around the world. A very critical piece of information when you're developing a vaccine against that disease. People will say, well, why are you developing a vaccine against invasive non-typhoidal salmonella? Well, there isn't much data on invasive non-typhoidal salmonella disease burden in low and middle income countries. the work that IVI did helped help people to understand the number of infections and the burden of disease and death imposed by invasive non-typhodal salmonella, justifying the work on the vaccine. What you see here are the 59 state parties and signatories to the IVI agreement and World Health Organization, as well as 16 countries that are in the process of joining IVI. So what you see in the state parties and signatories are the 55 or so organizations that have asked to join IVI. What you see in the last 16 are those countries that have applied to IVI, have been accepted by the board, and are still in the process of accessing. The headquarters of the International Vaccine Institute is located in Seoul in South Korea. In fact, we were the first international organization to be headquartered in South Korea. In 2022, we opened a regional office in Europe, shortly thereafter a country office in Austria. Last year, in the beginning of this year, we opened the regional office in Kigali, the African regional office. This will be followed later this year by a country office in Nairobi, in Kenya. Also last year, we formally signed the agreement with Hong Kong University to create the Hong Kong Jockey Club Global Health Institute, which is a three-way collaboration between IVI, Hong Kong University, and Cambridge University in the United Kingdom. In addition to these regional offices and laboratories and country offices, we have several of what we call collaborating centers. These are places, not official offices, but places set up by the collaboration, typically in places where IVI has had long-standing collaboration. So Bhutan, Burkina Faso, Ethiopia, Ghana, and Madagascar. And you can see them located. They're the countries that are in teal. Doing the work of vaccine development globally is, in fact, a global enterprise. You can see the little pinpricks that show you where the people at IVI are conducting work in discovery, development, delivery, and epidemiology and surveillance. You see them in Latin America, Sub-Saharan Africa, South Asia, and Southeast Asia. So really, the work that we do around the world in support of vaccine development for global health. This looks like a house, and in fact, the vision and mission of IVI are supported by four central pillars. The first around laboratory research, vaccine development, epidemiology and surveillance, and understanding real-world evidence of vaccine effectiveness. These support the strategic initiatives which are part of the 2022 to 2026 strategy. First, IVI needs to become bigger and better at doing discovery, development, and delivery. That is, amplifying the core and pursuing opportunistic actions that would allow us to more effectively or more efficiently develop vaccines for global health. The second is around international expansion, or what we used to call scaling the global footprint. In 2022, when the strategy was first put into place, IVI existed only as a headquarters in Seoul in South Korea, and you saw on the previous map that we now exist as official regional offices in Stockholm and Kigali, and then as country offices in Austria and Nairobi, with the opening of a Jockey Club office in Hong Kong. The Jockey Club laboratory in Hong Kong. So scaling the global footprint. And finally, around future-proofing governance. At the time in 2022, IVI's board was consisted of 10 at-large members and members with representatives from each of the funding countries. What we did in 2022 was we added 2 countries that were officially members of IVI, but were non-funding countries. Why? Because one of the things that we learned during the pandemic was that we had to give voice to countries that often don't have a voice in the development of vaccines or in the argument for access and equity in global health. This is a kind of complicated slide, but it really gets to what we're doing, the vaccines we're developing, and where along the value chain we work on these vaccines. So you can see, for instance, under cholera, IVI works in discovery, development, and delivery, and epidemiology and surveillance. So you see the IVI symbols in all of these. For some of them, you don't see IVI symbols everywhere. So for instance, Klebsiella pneumoniae is really only in the laboratory right now because that vaccine is still being developed. It's still in preclinical stage. Actually, we're still purifying proteins and polysaccharides from Klebsiella pneumoniae. What you see for Salmonella typhi, again, are IVI symbols throughout the vaccine value chain. IVI does not need to work on a vaccine that starts in our laboratory. Very importantly, we partner with organizations that need assistance at any stage of vaccine research and development. And you can see that better on the next slide, which are the viral diseases that we work on. SK Bioscience, which had a World Health Organization pre-qualified COVID-19 vaccine, used IVI primarily to do clinical, the laboratory testing of the immunogenicity of the product, and also to help execute the trial outside of Korea, so in countries around the world. And we can do that. It doesn't need to start in our laboratory. IVI can help at any stage in vaccine research and development. One of the things at the bottom is something I'd like to highlight, MAP, which stands for microarray patch. This is a technology that's been prioritized by Gavi, the Vaccine Alliance, and the World Health Organization. We apply the vaccines to a patch and can deliver them on a patch rather than through needle and syringe. This simplifies delivery because now you don't necessarily need a nurse to be able to apply the patch and deliver the vaccine. But also, patches have in general, are more heat stable, so you can actually store vaccines at room temperature, and might theoretically be easier to transport because the volume of transport is lower and because of the lack of a cold chain requirement. So patches could be a very important technology of the future, and IVI is working with several companies on different versions of patches for vaccination. When we talk about IVI's vaccines, we really have in mind three. Vaccine number 1, which is the oral cholera vaccine, vaccine number 2, which is typhoid conjugate, and vaccine number 3, which is a vaccine for non-typhoidal salmonella. So these are vaccines that originated in IVI laboratories and have now resulted in progress in the case of cholera vaccine through 5 pre-qualifications. In the case of typhoid conjugate vaccine, the pre-qualification of the SK Bioscience typhoid conjugate vaccine, with the biopharma typhoid conjugate vaccine, again, tech transferred from IVI, receiving approval from the regulatory authority in Indonesia, known as BPOM, and with World Health Organization submission for World Health Organization pre-qualification pending. Non-typhoidal salmonella vaccine number 3 is a vaccine that is ready to enter human clinical trials. We're currently looking for funding to do that. We've started work on vaccine number 4, which will be a vaccine against Klebsiella pneumoniae, and are currently trying to decide what vaccine number 5 will be. So these are all vaccines that are in development. In the case of vaccines 4 and 5, still in either conceptual stage, as in vaccine number 5, or in a phase of purifying proteins and polysaccharides, as the Klebsiella vaccine. But we also work to strengthen the ability of low and middle income countries to have vaccine development or vaccine manufacturing within their borders. This includes training and education, work with global health strategic groups, and then technology transfer. So in training and education, the 1-week IVI vaccinology course has trained over 5,000 people around the world. This year it was offered in Seoul in South Korea, in Stockholm, and in Kigali simultaneously. So it's offered in the beginning of September and the beginning of September, and an announcement goes out in the summer asking for students who might apply for scholarships. A second training activity is known as GTHB, or Global Training Hub Biomanufacturing. This is a World Health Organization, Korean Ministry of Health and Welfare joint activity that is the first step in the WHO vaccine manufacturing curriculum. So this is the introduction to vaccine research and development. That is the first course. It's 2 weeks long. And the second course, which is 3 weeks long, is a course in quality, in what we call the GXPs, Good Clinical, Laboratory, and Manufacturing Practices. So far, over 900 students have come through these 2 courses. In fact, the GXP course in 2025 is ongoing now. Working with GHSA globally, with partner countries around the world, and then also with ASEAN and the ASEAN Vaccine Security and Self-Reliance Network, And this is a collaboration with not only the ASEAN countries, but primarily with the National Vaccine Institute of Thailand. Technology transfer is at the core of what we do. We've done 14 technology transfers of 6 different vaccines to developing country manufacturers around the world. Within Korea, IVI has worked to strengthen the biotechnology ecosystem of the country. This includes work with the Korean government on something called Vaccine 3.0, which was a $300 million program to build the Vaccine Research Center at the Korea National Institute of Health, and to create 2 pilot plants, one for bacterial vaccines, one for viral vaccines, and then to work to develop a GLP laboratory at one of the pilot manufacturing plants. And IVI planned with the Korean government and operates one of the laboratories at the plant. In 2018, we worked with the Korean government and the Gates Foundation to create the Right Foundation, Research, Innovation, and Global Health Technologies, which is an innovative new idea which allows 50% Korean government funding, 25% Gates funding, and 25% funding from companies to be leveraged against other funding to develop vaccines, drugs, and diagnostics for global health. So many people understand that there's a big gap between what a professor has in a university and an actual product that you can sell and will be useful for global health. The Right Foundation provides funding to take things out of the laboratory into manufacturing, into clinical testing, and sometimes through approval, so that several of the products that the Right Foundation has funded, diagnostics and vaccines, are either have received WHO pre-qualification or are soon to be set forward on the pathway towards WHO pre-qualification. So effectively developing products for global health, at the same time developing those biotechnology companies, supporting the biotechnology ecosystem around innovation and translating innovation into products, which are really critical. And when we think about COVID, was there a gap in low and middle income countries between being able to hope for a COVID vaccine or a COVID diagnostic and actually being able to manufacture it within the country? The Right Foundation helps small companies to develop these skill sets to have the manufacturing capacity there when it's needed. In 2020, IVI worked with the Korean government around these 3 vaccine R&D hubs. So again, the Korean government had wanted to become 80% self-sufficient in vaccines by 2030 and the number 5 vaccine manufacturer. Well, in order to have that kind of manufacturing capacity, you need to have people who are trained in biomanufacturing or in biotechnology. And so the R&D hubs, which are several $100 million grants over 5 to 7 years, were designed to develop new innovative technology, cutting-edge technology, to train people to be technicians, to train people to think and develop products as postgraduates or as PhD level scientists. A really important part of their human resource development necessary, and IVI helped with the initial development of the concept around the R&D hubs, and then advises several of the hubs and works and partners with them on the development of other vaccines. So again, very involved in the generation of a strong ecosystem to support sustainable manufacturing. And finally, in 2021, IVI worked with the government of Korea, and this would be, IVI staff served on several of the important government committees that were put together to create the K-Vaccine Hub. The K-Vaccine Hub is a remarkable attempt to build manufacturing capacity in Korea, $2 billion worth of support for developing vaccine manufacturing. But importantly, and this is what IVI is doing, IVI's role in this actual role, was around the training hub. The GTHB, Global Training Hub Biomanufacturing, is actually located funding-wise within the original K-Vaccine Hub. So IVI has partnered with Korea to build an ecosystem around biomanufacturing and biotechnology that strengthens the country's ability to respond during pandemics. This means that IVI goes from the development of vaccine products from need through innovation to impact. And you can look at the cholera vaccine. There was a need for a cholera vaccine that could be used in global health settings where you didn't have access to clean water, where you needed significant quantities and doses very quickly. So taking that need, we developed the oral cholera vaccine. Technology transferred it to several companies around the world. 200 million doses of oral cholera vaccine have been dispensed now. between 200 and 300,000 lives have been saved, estimated, because of the availability of those doses of cholera vaccine. Similarly, for COVID-19, there was a need for COVID-19 vaccine. IVI worked with SK Bioscience, which got information from the Institute for Protein Design, and the first AI-generated approved vaccine was the one that we worked on with SK. So AI, we talk about AI and how AI is going to transform vaccines and vaccine development, vaccine research and development, in our entire lives. But in fact, the COVID-19 vaccine from SK was the first product that was approved not only by the Korean government, but by MHRA and by the World Health Organization, and that was AI generated. So again, the intersection between needs and impact is innovation. And we work with innovation, and you can see the areas where we work, to develop products that will have an impact in global health. Thank you very much. And there are IVI staff who are attending the ISV meeting. If you have a question about IVI, write to us, contact us, or speak to some of the IVI staff who are attending the meeting. Thank you very much.
Now please welcome Professor Alexander Bukreyev from University of Texas Medical Branch at Galveston. Professor Bukreyev is a distinguished expert in virology and vaccinology with extensive research on emerging viral diseases and respiratory infections. His pioneering work on Ebola virus and respiratory syncytial virus has made significant contributions to global vaccine science. His presentation today is titled Vaccine Research at the University of Texas Medical Branch at Galveston and Galveston National Laboratory.
Hello, my name is Alex Bukreyev. I am professor of virology and vaccinology at the University of Texas Medical Branch at Galveston. I'm also associate director of the University of Texas Medical Branch Center for Biodefense and Emerging Infectious Diseases. Today, I'm going to show you to talk about vaccine research at the university at UTMB. Let me share my screen. Okay, this is our university, UTMB. It's located in Galveston, coastal city of Galveston. This is a view on a campus. And UTMB is a component of the Texas Medical Center located in center of Houston. This Texas Medical Center includes multiple institutions with a gross domestic product of $25 billion US. And even though UTMB is located about 80 kilometers from the main campus, it is a component of this center. So this picture illustrates UTMB capabilities for vaccine research. We have Center for Biodefense and Emerging Infectious Diseases and Sealy Institute for Vaccine Sciences. The Sealy Institute for Vaccine Sciences was established in 2001 as a Sealy Center for Vaccine Development. Later, in 2018, it was converted to Sealy Institute for Vaccine Scientists. And the institute support and guide development of prophylactic and therapeutic vaccines at UTMB. The vaccine-related research at UTMB involves discovery, basic and applied research, preclinical development, clinical trials, as well as education. So you can see
basic science is typically run by faculty members, and then preclinical studies. Regarding preclinical studies, we have now a contract with National Institute of Allergy and Infectious Diseases for preclinical testing of vaccine, not only developed at UTMB, but also by other entities in the United States. Some of these studies are performed guided under guidance of the Office of Regulated Non-Clinical Studies. So these are regulated studies. Clinical trials program includes phases 1 through 4, and also we have a lot of educational activities. Okay, this cartoon illustrates the various steps of the development of vaccine, from basic research to proof of concept to concept validation, and to preclinical GLP studies toward clinical trials or FDA approval through animal rule. This is the current contract with the National Institute of Allergy and Infectious Diseases, which I already mentioned. It goes through 2021 and includes several tasks, from small animal models to non-human primates, animal replacement, and non-traditional models, and generation and distribution of specialized reagents. We have a lot of experience in clinical trials, which are being run by Dr. Richard Ropp, which is director of the clinical trial program, as well as currently acting director of the Sealy Institute for Vaccine Sciences. And from the first trial in 2001 to the current trials, this slide shows multiple trials which were run by UTMB from 2001 to present time. They included, as you can see, phases 1 through 4, and includes vaccine against viral and bacterial pathogens, and includes pediatric, adult, and older adult participants. And importantly, there are 3 important books in vaccinology, in the field of vaccinology, which were edited by members of the Sealy Institute for Vaccine Sciences. Okay, this was about the Institute for Vaccine Scientists, and the next few pictures will illustrate our capability for preclinical testing of vaccine against emerging viral infections or emerging infections in general, which require biocontainment. So this is Galveston National Laboratory. So the laboratory is supported by the National Institute of Health, has a major network of collaborators in endemic regions for most biothreat viruses, particularly in South America and Africa. And we have experience in aerobiology, vector-borne transmission, including BSL-4 and other areas. Importantly, this Galveston National Laboratory is a home for NIH-funded World Reference Center for Emerging Viruses and Arboviruses, with over 6,000 strains available from around the world. So this is the layout of the Galveston National Laboratory. It has laboratories from the biosafety level 2 level, animal biosafety level 3, biosafety level 4, and biosafety level 3+. And here are mechanical floors. Also, this red spot is a Robert Schoep biosafety level 4 laboratory. These 2 labs, the buffer corridors, are connected by a corridor. This is again the layout of the biosafety level 4 maximum containment laboratory, and this is how the facility look. This is example of biosafety level 2 laboratory, biosafety level 3, and biosafety level 4. Again, more pictures. And this is a technical floor. This is air handling and HEPA filtration. So these boxes are filled with HEPA filter, and these pipes, this is for air support to the BSL-4 laboratory, which are located in the next floor down. And this is backup generators where they're tested on a monthly basis. And these are effluent waste system, which is located just next level under the BSL-4 lab. These are huge autoclaves. The Galveston National Laboratory has service division, air biology, assay development, experimental pathology, imaging, insectaries, and preclinical studies. This cartoon illustrates specialized equipment available in the Galveston National Laboratory. And these are animal species currently supported, animal species for preclinical testing of vaccines. Okay, this block of slides was about our capabilities for testing of vaccine in biocontainment for challenge studies. And the next couple of slides is capabilities of my laboratory. So I am involved in vaccine research for almost 3 decades, 15 years at UTMB, and before that, also about 15 years at the National Institute of Health in Bethesda, Maryland. One line of vaccine research in my lab at UTMB is collaboration with Moderna, which started in 2017. This collaboration resulted in, as you can see, these publications on mRNA vaccine against Ebola virus, Marburg virus, SARS-CoV-2. As an example, this paper, it's on SARS-CoV-2, published in 2021. Before it was published, the data which were included in this paper were generated in this paper were included in the Moderna's investigational new drug and biological license applications to the FDA. Of course, along with the data generated at the NIH and some other laboratories, which eventually led to approval of the vaccine, mRNA vaccine against COVID-19. As a separate line of research, we develop vaccine in our lab from scratch. And this is mRNA vaccine against Andes virus. This is a highly pathogenic South American virus. And this was published in summer 2024. Another vaccine is mRNA vaccine against Rift Valley fever virus. We've shown that a single 1 microgram dose completely protects mice against death and disease caused by the virus. And we have several more vaccines developed from scratch, which is in the pipeline in our lab being tested. As a separate vaccine platform, we also developed circular RNA. So circular RNA was initially, circularization was optimized by 2 laboratories. These are publications at Massachusetts Institute of Technology and Stanford University. And it was demonstrated that circular RNA provide expression in vivo for a longer period of time, roughly 7 days, compared to linear mRNA, presumably due to resistant to exonucleases. So that is the reason we started these studies with circular RNA based on publication from Shan lab at La Jolla Institute of Immunology in California and some other labs. Longer duration of antigen expression results in more durable antibody response. So we generated our own platform circular RNA platform, which was the patent was filed earlier this year, and we have several circular RNA-based vaccine in the pipeline now being developed, or some of them are being preclinical tested. And this is the last slide. This is my laboratory at UTNB. Beside vaccine, we also work on investigation of dysregulated immune response to Ebola virus at the epigenetic level. We work with multiple highly pathogenic viruses: Ebola, Marburg, Lassa, Rift Valley fever virus, New World and Old World hantaviruses, Crimean Congo hemorrhagic fever, respiratory syncytial virus, and can work with other viruses causing acute infection and which require biocontainment. Thank you for your attention.
Now let's welcome Yoko Oi from the Chiba University Synergy Institute of Futuristic Mucosal Vaccine Research and Development. This program is very well known on mucosal vaccine research. It is established by Professor Hiroshi Kiyono as the world-renowned authority in mucosal immunology and vaccine research. His presentation today will describe their latest work in mucosal vaccine research and development. Here is the talk from Chiba University.
Hello, my name is Yoko Oe. I'm working as a university research administrator at Chiba University. We are honored to be a part of this important congress and to share our research efforts and future directions with a distinguished colleague from all over the world. I would like to introduce an important institute at Chiba University, Synergy Institute for Futuristic Mucosal Vaccine Research and Development, which calls CSIMBA. Under the leadership of Director Dr. Hiroshi Kiyono, CSIMBA work as one team to advance cutting-edge research in mucosal immunology and vaccine innovation. This slide shows the overview structure of the Japan Agency for Medical Research and Development, AMED, SCARDUP project for establishment of a Japan initiative for world-leading vaccine research and development centers. Based on the strategy for strengthening the vaccine development and production system, and aiming for the achievement of Japanese-made vaccines, world-leading vaccine research and development institute are set up. The University of Tokyo has been selected as a flagship institute, while Chiba University was selected as one of the Synergy Institutes. These institutes Synergy Institutes work closely with the flagship institute while leveraging their unique strength, creating the synergic effect across the network. To promote sustainable mucosal vaccine development, we have established the international development structures with the University of California, San Diego, the La Jolla Institute of Immunology, as well as domestic research institutes. In addition, we have partnered with 11 companies and 6 research institutes to create effective, safe, and secure mucosal vaccine. Additionally, we have launched the vaccinology and the infectious disease courses to train next generation of global vaccine experts in a seamless and interdisciplinary manner. CSIMBA focuses on deepening our understanding of a mucosal immune system, and advancing the development of innovative mucosal vaccines. Next, I would like to introduce the structure of CICIMBA. In terms of implementation structure, under the leadership of Director Dr. Hiroshi Kiyono, the Institute is led by 4 deputy directors and a team of excellent 16 PIs, including 10 basic researchers from Chiba University and UC San Diego, 5 clinical researchers, and 1 researcher from industry, who collaborate, drive research and development activities. Regarding the industry collaboration, the Institute partners with 2 vaccine-related companies, 3 university-launched venture specializing in a vaccine and immunology, 1 company engaged in a plant-based green vaccine production, and 5 companies specialize in a vaccine delivery and adjuvants. As for partner research institutions, we will further strengthen ongoing collaboration with the 6 university and research institutes, with which joint research is already underway, and enabling us to establish strong and effective partnership. Our CICIMBA vision is save lives and livelihood by safe and friendly mucosal vaccines. Our missions include creating pain-free and worry-free mucosal vaccines, preventing the pathogen invasion, and also developing mucosal vaccine with a Society for Resting Vaccine Trust. CICIMBA structure is built on the integrated 3-level strategic research and development system. In next slide, I will explain the detail. We operate through this integrated 3-level strategic research and development system, which brings together universities, companies, and the researchers under the shared vision of a mucosal vaccine R&D. Participating members are able to clearly understand how their individual research fits into the overall mission. Together with a director, deputy director, and PIs, we coordinate and advance research across levels in a cohesive and strategic manner. Our structure consists of 3 interconnected levels. 1 is a new knowledge, which is elucidating mechanism of innate and adaptive immune induction and the regulation in a mucosal immunity. 2nd, novel technology, which is conducting the research and development to identify and refine the candidates for mucosal vaccines and adjuvant by optimizing using AI and data science. And 3rd, technology integration, which is confirming the efficacy of developed mucosal vaccines, adjuvant candidates, and advancing and conducting the human POC clinical trials for mucosal vaccines. All 3 levels interact in the continuous cycle, enabling the sustainable generation of a scientific knowledge and its translation into the real-world implementation. So through this integrated 3-level strategic research and development system, we promote the research and development of a safe and reliable mucosal vaccines. We discover the new knowledge of the respiratory and digestive mucosal immune system, and then novel modality for oral and nasal vaccines, and two new delivery system and adjuvant. In our institute, we focus on the development of oral and nasal vaccines, which have a unique ability to induce the secretory IgA antibodies at the mucosal surface, where pathogen invade in addition to systemic immunity. Mucosal vaccines are highly promising as a preventive vaccine capable of both preventing infections and reducing the severity of a disease. We are developing oral vaccines aimed at preventing intestinal infections such as enterovirus, norovirus, Vibrio cholera through mucosal immunity in the oral, pharyngeal, and the intestinal tract. Cold chain and the device-free oral vaccine Mucorice is a rice-based oral vaccine for cholera and travels diarrhea, in which the non-toxic B chain CTB antigen of a cholera toxin is expressed on the rice. We are developing also nasal vaccine targeting the respiratory tract infection such as SARS-CoV-2, pneumococcus, and influenza, and RSV, using cationic nanogel-based antigen delivery system. In next slides, I'll explain detail of Mucorice and cationic nanogel, DDS. I would like to explain about the Mucorice. As a prototype, we have developed a rice-expressing B subunit of a cholera toxin, an important binding molecule for the CT receptor expressed on the human cells, including the gut epithelial cells for the initiation of the diarrhea symptom. As shown in the slide, CTB gene has been inserted in the rice protein expression system. An electron microscopic analysis using the gold particle label CTB-specific antibody confirmed the expression and accumulation of CTB in the rice protein body. These CTB-expressed rice are named as Mucorice CTB. As illustrated in this slide, CTB expressed in the protein body are protected from the digestive enzyme, such as pepsin, and taken up by the antigen sampling cells, M cells, located in the epithelium of a gult or Peyer's patch. So the rice protein body are serving as a natural capsule for delivering the vaccine antigen to the gut immune system for the induction of an antigen-specific immune response. This project arose out of a unique blend of expertise in medical science, agriculture, and engineering. We were inspired by the fact that the rice can be stored at room temperature for a long time. Mucorice offers a safe, cold chain, and a needle-free delivery system, and it does away with the need for syringe and expensive refrigeration. So this is going to be an important consideration, particularly in the developing countries. Next, I would like to explain the cationic nanogel nasal immunization. We developed the nanometer size hydrogel, a nanogel, consisting of a cationic cholesterol group bearing Poland, called CCHP, we found that our CCHP nanogel is a safe and effective nasal vaccine delivery system that can be used to stimulate the immune system of the nasal mucosal by using a variety of subunit antigen. Because of its cationic property, CCHP nanogel show persistent attachment to the surface of the negatively charged nasal mucosa, leading to the prolonged release of antigen to the antigen sampling and presenting system of the nasal mucosa. This slide shows the evidence of efficient antigen uptake by antigen-presenting cells such as macrophage and dendritic cells. Induced protective immunity against the antigen was observed. So, with a strong understanding and support of the Chiba University, and under the leadership of a director, Dr. Hiroshi Kiyono, we envision a society where infectious disease are controlled through the mucosal vaccines within the next 10 years. Through our integrated 3-level strategic research and development system, and by fostering the next generation of the skilled vaccine professionals, we aim to contribute to a sustainable mucosal vaccine development platform in alignment with the SDGs, and also a safe and worry-free vaccination society. Together as a one team, Chiba University and UC San Diego, we are united in our commitment to develop the mucosal vaccines as a global standard from Sisimba at Chiba University to the world. Thank you very much for your attention.
Hello, I am Denise Doolan, Vice President and Immediate Past President of the International Society for Vaccines. Now let's welcome Dr. Fran van Heuverswyn from the Immunity for Health Belgium. Dr. van Heuverswyn has extensive experience in vaccine innovation and public-private partnerships. She has been dedicated to fostering collaboration amongst academia, industry and public health sectors to advance vaccine development and accessibility. Her presentation today is titled Immunity for Health, a Belgian catalyst for public-private partnerships in vaccines. Welcome.
Good afternoon, everyone, and thank you to ISV for inviting me to present the Belgian vaccine ecosystem and the role of our organization, Immunity for Health. So my name is Fran van Heuverswyn, and it's a real pleasure to share how Belgium, despite its small size but complex structure, has built a powerful and collaborative network driving vaccine research, clinical development, and innovation. So here is what I will cover today. So first of all, I will start with why vaccines matter. I think most of all are, of course, aware about their importance, and also how they have shaped public health globally. Then I will move to why Belgium and explaining what makes our country a leading hub for vaccine research, clinical trials, and production. And finally, I will introduce Immunity for Health. It's our cluster organization that connects and strengthens this ecosystem through collaboration and innovation. So why vaccines? So vaccines are among the greatest achievements of modern medicine, saving over 154 million lives to date and preventing countless more cases of severe diseases, as published in this Nature article. However, in this article, public health experts warn also that misinformation and funding cuts could heavily affect the vaccination rates. So continued investment, collaboration, and trust in science are essential if we want to keep vaccines to save lives. Child vaccination programs are the best proof of how effective vaccines can be. So diseases that once caused enormous suffering, like measles, tetanus or pertussis, have been largely controlled or even eliminated thanks to sustained immunization efforts. These charts illustrate that the success of vaccines was really great. So we see over time that when vaccination is introduced, we see a dramatically drop in the incidence of disease, but also in the mortality rates. As for measles here in the US, also for polio or other diseases. So now, that brings us to our next part. Why Belgium? So despite being such a small country, Belgium truly punches above its weight. We're amongst Europe's top 3 when it comes to biopharmaceutical R&D investments, also for patent applications and for clinical trials, which is shown on this slide. So what makes our vaccine ecosystem so unique? So Belgium is a real world leader in vaccine research, development, manufacturing, clinical studies, and the distribution. So our universities are internationally recognized. So 9 of the 11 universities are featured in the QS World University Rankings of 2025, and even 7 of them are within the top 500. We also have 7 university hospitals, which are in close distance from our academia, so the university and academic labs, which allows a rapid translation from the discovery to the clinical applications. Belgium also counts 14 biotech incubators, and we have the major pharmaceutical companies such as GSK and Pfizer in Belgium, with the long-standing R&D or production site here in Belgium. We also have a very strong regulatory system led by the Federal Agency for Medicines and Health Products, ensuring both scientific quality and patient safety. And finally, we shouldn't forget our excellence in pharmalogistics. So Belgium is home to Brussels Airport, which is Europe's most important pharmaceutical hub. During the COVID pandemic, over 1 billion of vaccine doses were imported and exported through this network. So all of these elements together, so the strong academia, innovative industry, efficient regulation, and world-class logistics, make Belgium an important country with a globally significant vaccine ecosystem. So Belgium excellence is also reflected in its clinical trial network. So this slide gives an overview of the main human vaccine clinical trial centers in Belgium, highlighting both academic, also some private sites. So here at Ghent University, the Center of Vaccinology has over 37 years of experience in vaccine development and evaluation, combining clinical and laboratory work. At the University of Antwerp, the Center for Evaluation of Vaccination has led more than 330 trials over the 25 last years. And recently they opened Vaccinopolis, a unique European facility for controlled human infection studies. Also in Antwerp is the Institute of Tropical Medicine, and they launched their clinical trial center in 2022, focusing on vaccines, medicines, and diagnostics for infectious diseases. In Wallonia, new academic initiatives are expanding this national research capacity. So the centers which I mentioned before were all in the northern region of Belgium, so in the Flanders, in Flanders, and then in the southern region, the French-speaking region, new initiatives are also coming up. And then, of course, we also benefit from strong private partners. 2 of them are mentioned on the slides, but of course there are many more. Miklinas and SGS are two members of our organization. So Miklinas is in Mechelen, and it's a network of general practitioners specialized in phase 2 and 3 vaccine trials, but also other conditions. And then we have SGS here on top. So the SGS Clinical Pharmacology Unit conducts early phase studies, and they have facilities for over 100 volunteers. So together, these centers, they form a highly connected national network covering every phase of vaccine testing and complemented with the possibility to perform CHIM studies. So this is making Belgium one of Europe's most efficient locations for vaccine research. Two centers stand out in particular. I already mentioned Vaccinopolis, which is part of the University of Antwerp. It's a big facility. You can see it also on the picture. A facility of 6,000 square meters. It includes a quarantine unit for controlled human infection models. It also has outpatient vaccine trial units and high-level biosafety laboratories. So the University of Antwerp has a joint initiative, so an important collaboration with the European Pathogen Institute for Vaccinology, which is at the ULB in Brussels. And this facility, has a really strong expertise in immunology and microbiology. So together they form a unique academic partnership that enhances Belgium, but also Europe's preparedness for emerging infectious diseases. This graph is just to show you some figures. So this graph shows you the total number of prophylactic vaccine clinical trials performed in the last 5 years for the top 10 European countries. So here we can see that Belgium is doing very well. And if we look to the next graph, where the data are shown as the number of clinical trials per capita. So we see that for both parameters, so both graphs, Belgium is number 1, so demonstrating we are one of the top performing countries in Europe. This is reflecting our unmatched density of vaccine expertise, but also the infrastructure. This slide, this slide shows you also our contribution on a European level. There is a new initiative, the European Vaccine Hub for Pandemic Readiness. It's recently launched, and the aim of this project is to create a permanent agile structure that connects European partners that come up that they can contribute with different technologies, with containment labs, if they have them, serology, immunology capacities. So different techniques and different expertise are welcome within this project. And Belgium is actively involved in this initiative, with Vaccinopolis as the lead of pillar 3 of this project. So they are responsible for the clinical research part. So the Belgian contribution to this European project builds on the long-standing experience in vaccine development and also on the public-private collaborations that we have. So it's a perfect example on how our national strengths can translate into European, but even global impact. So this brings me to our organization, so Immunity for Health. Immunity for Health is a non-profit organization. It's a health cluster established in 2016. And originally, we were known as Flanders Vaccine. You can still see it here on the pictures with a banner with our old logo. Our new logo is here mentioned or shown on different slides. So to strengthen our visibility abroad and to better position our expertise in both vaccines, but also immunotherapy worldwide, we decided very recently, so a couple of weeks ago, to renew our identity to Immunity for Health. And our mission is to stimulate end-to-end development of vaccines and immunotherapeutics by building on the strengths, the expertise that is already present in the human, but also in the animal R&D sector. So the One Health concept is also very important to us. Our objectives, objectives are very clear. So we want to support innovation in the sector. We want to bridge the gap between academic research and industry. We want to stimulate partnerships across human and animal health. We also want to create a positive environment for vaccine clinical trials, to create a strong network, and to promote this network as a leading ecosystem for vaccine development. Our strength lays not only in our projects, but also in the people who guide our work, of course. Here you can see our board of directors. It's a diverse group representing academia, industry, and biotech. So we have representatives of the 5 Flemish universities, and their combined expertise ensures we remain relevant, balanced, and responsive to the needs of all our members. So, and of course, the true strength of our organization comes from our members and partners. So currently we have about 50 member organizations joining our network. We have 3 categories, so with knowledge and public institutions, and then private companies, so the companies that perform R&D, and the service companies. So together we form a multidisciplinary ecosystem that covers the entire value chain. So from fundamental research to clinical development, production, and delivery. So what are we doing? So we organize a wide range of activities to foster dialogue and collaboration. So our annual event takes mostly place in October. And the name of our annual event was also Immunity for Health. So this is a bit the reason why we have chosen that name also for our organization, because it's already familiar to a lot of people. And this event covers every year another theme. This can range from vaccine hesitancy, One Health topics, or related to clinical trials, AI, and vaccine development, adult immunization. So a very broad range of important topics. Also for our symposia, workshops, and webinars, they are on emerging topics and often in collaboration with other partners. And we also organize elevator pitch sessions, which help early stage researchers and companies to showcase their innovative technologies to potential partners. Our organization also plays an active role in internationalization. So we have contributed to projects such as the European Transvac DS project, which strengthens Europe's vaccine R&D infrastructure. And we also support international missions and delegations that visit our country. And our latest initiative is in collaboration with our Flemish government and the WHO and Medicine Patent Pool, and is focused on strengthening health security and pandemic preparedness in Africa. So through these efforts, we ensure that our Belgian expertise has also a global impact. Next slide. So what can Immunity for Health offer for you or for our members? So being part of our organization means joining a community that connects, collaborates, and innovates. So we help our members to find the right partners for their projects, help them to inform them about funding opportunities, and also enhance their visibility. So in short, we create the right environment for ITs to grow into impactful health solutions. So how can we collaborate? So there are a couple of ways to collaborate with us. So of course you can join as a member, we can become a strategic partner to help shape our activities and to advise us on our strategy. Or you can also sponsor or partner with us on one of our events. And we also design custom-made events and collaborations so tailored to your needs or the needs of our partners. And I also want to thank our strategic partners, MSD, GSK, Sanofi, and Pfizer, for their valuable support and advice. So this brings me to my last slide with the conclusions. So to conclude, we can say that Belgium is really internationally recognized for its excellence in vaccine R&D, but also in the conduct of clinical trials. And our success is built on a unique collaboration between the academia, the industry, and the government. And through our organization, Immunity for Health, we aim to strengthen this ecosystem and to represent it on an international stage. And ultimately, collaboration is really key, not only within our regions, but also across Europe and even beyond, if we want to remain competitive, globally competitive and ready for the next health challenges. So this was my last slide, and I thank you for your attention.
The next presentation is by Dr. Baik Seong, who is professor of biotechnology at Yonsei University, Korea. He's also director of Vaccine Translation Research Center and director of Vaccine Initiative Technology Alliance Korea. Dr. Seong will be talking about vaccine self-sufficiency in Korea. Please, Dr. Seong.
Hello, everyone. My name is Baik Seong, a Director General of Vaccine Innovative Technology Alliance, Korea, and I would like to use this privileged opportunity to briefly report on our current efforts for establishing vaccine self-sufficiency in Korea. Our organization is a Korea government R&D initiative started early 2020 under the support of Ministry of Health and Welfare. Korea has long suffered from endemic and epidemic outbreaks of infectious disease. Actually, the lack of domestic production of vaccines of NIP programs has led us to rely on the supply chain overseas, rendering Korea vulnerable to vaccine shortage during the unexpected outbreaks. So, to overcome these issues, we established a translational research from preclinical to clinical evaluation of vaccine candidates by collaboration with clinical research organizations, industries, including pharmas and biotechs, and utilizing GMP-level production facilities, and collaborating with international organizations like IVI, in support with the government agencies like Korea Disease Control and Prevention agencies, and also by consultation with the regulatory authorities like Ministry of Food and Drug Safety. So our mission is to secure vaccine sovereignty to strengthen public health and establish a stable infrastructure of vaccine development. We also have a vision to emerge as a vaccine G5 country later on, with the goal of the expediting domestic production, of increasing the self-sufficiency of NIP vaccines, and developing vaccine candidates of global needs. In 10 years of duration, with the total budget about over $200 million from the government support. So currently, the Korea, the vaccine production is only less than 40%. We want to increase it up to 80% after successful completion of the project. And with NIP vaccines, we have 4 different target areas: Japanese encephalitis, DTaP, and hepatitis A virus, and human papillomavirus as well. Second mandate is globalization. Currently, the overall global vaccine map, there is no Korea visible here, but we hope to emerge as a visible vaccine producer after the completion of this project. We also have to develop technical innovations, not only on delivery platforms, but also adjuvant that could be applied for new vaccines and improving traditional vaccines as well. In the current vaccine map that have established over 70 years, the VITAL is positioned to dedicate development of NIP vaccines and in part the unmanaged vaccines. Whereas government organizations is more focused on national security program, especially with the future pandemic areas as listed by the WHO priority list of infectious disease. In terms of technical innovation,
Hello, I am Denise Doolan, Vice President and Immediate Past President of the International Society for Vaccines. Now, let's welcome Dr. Madeline Clark from the International Veterinary Vaccinology Network. Dr. Clark has been dedicated to promoting international collaboration in veterinary vaccinology, advancing vaccine research and development to combat livestock diseases. She plays a key role in fostering partnerships and capacity building, especially in low and middle income countries. Her presentation today is titled The International Veterinary Vaccinology Network, Working Together to Develop Vaccines for Livestock Diseases. Welcome.
Okay, hello everyone, and thank you to the International Society for Vaccines for inviting myself and the International Veterinary Vaccinology Network to come and provide an online presentation of what IVVN does. I think it's a fantastic idea to prepare these recordings for a virtual conference alongside your wider in-person conference, and I really hope everyone attending has a fantastic few days. Very, very sorry to not be with you all. Okay, so my name's Madeline. I am the network manager for the International Veterinary Vaccinology Network. So IVVN is an open access platform for the global veterinary vaccinology community. Its vision is to create a multidisciplinary and interconnected network to tackle the challenges of developing vaccines for livestock and zoonotic diseases, particularly in low and middle income countries. So IVVN is funded by the Medical Research Council and the Biotechnology and Biosciences Research Council in the UK, but we are a global entity. So what do we do? So as I said, we're made up of a global community of key stakeholders working in veterinary vaccinology, dedicated to developing vaccines for high consequence livestock diseases. Our chief aims are really to support this network of members and facilitate collaborations to address the key challenges impeding vaccine development. And we do this through a variety of activities. So we support facilitating opportunities for networking through our international conferences we've held and workshops. We also support collaborative funding through small pump prime grants and laboratory exchanges. We support the training of early career researchers to gain new knowledge, but also help them become leaders in the field. And we promote gender equality in veterinary vaccinology through supporting women fellowship schemes, as well as our African Schools Outreach Programme. A central aim of the network is to be a central knowledge hub for the community. So we've got an interactive website where we request members to send in resources, events, opportunities like job postings, publication and news in the field that we update on our website, along with providing information on the activities that we directly run. And we also have a number of other communication platforms. We run a monthly webinar, we run a monthly newsletter, and of course we're active on LinkedIn. So the community so far is made up of 2,472 members across 93 countries. You can see on the map where these members are based. This can be found on our website. So on our website, we have this members directory. So you're able to join as a member through this QR code, and really the members directory it's a searchable database for people, if you're looking for people working on specific pathogens or hosts or a particular stage of vaccine development. Members input that information like their biography and what their research interests are, along with external sites like LinkedIn. And it provides that sort of easy way of being able to find other people working in the area and a chance to connect with them. As I said before, we've got a strong focus on supporting early career researchers. We've done this in the past through travel bursaries to conferences. We've supported a number of researchers to come to ISV before. We support lab exchanges. And then this year we funded the IVVN Career Development Fellowship Scheme. So we awarded 11 researchers around the globe an 18-month fellowship. It doesn't come with any grant funding, but it's there to support their career development. And so we've provided a sort of a curriculum, as you were, of supporting and facilitating peer-to-peer mentoring, where they decide what topics are important to them and they want to talk about and want to hear others' experiences, be it within the cohort or bringing in specialised people to discuss those. Journal clubs, we've got an external consultant providing leadership coaching to the cohort. Specific training, so again we ask the cohort what they would like training on. An example is how do you work more closely with industry? And we also provide mentorship from experts in the field, and this is in collaboration with the Plowright One Health mentoring scheme. And so, although they're a small cohort of the Veterinary Vaccinology Network, they're also part of this larger One Health mentoring scheme. So it's a wider network of early career researchers and providing support to them. And we're really looking forward to seeing how they develop in their careers going forward. As I said before, our flagship networking activity is our conferences. We've been running for the last 7 years, and of course we've had a global pandemic in that time, so we haven't held conferences every year, but we have held conferences in Nairobi, London, and last year in Ho Chi Minh City in Vietnam. And this is a real chance for the community to come together in person. We have provided recordings of the conferences as well. come together to hear about the latest research, but also to hear from other stakeholders like industry, like policymakers, hear about what the current state of control of certain diseases are in those regions. And along with the conferences, we always try to run some topic-specific workshops as well. So sometimes these are often geared up for early career researchers. So example might be grant writing, but we've also done some interesting other ones as well, like ethics in animal research, disease-specific workshops. We've done one on epidemiology and economics. And it's really members coming to us saying they would like to run this type of workshop, and we are here to help facilitate and organise the logistics of that. Another large flagship activity for us has been funding these small pump prime grants. So they're one-year projects of up to £100,000 with a key aim to address the key bottlenecks preventing the development of vaccines for livestock and zoonotic diseases in low and middle income countries. So this could be sort of a pilot piece of work that people need to do before they can, submit a larger 3-year application, or it could be a piece of work that industry really needs to happen before they can take up that research into vaccine development. So far we've run 5 rounds of this and 17 projects. We've currently got 18 published papers out of these projects. We've had some projects that have been taken up by industry, and it's also been really great to see how these projects have helped people's careers progress, some of them into like tenure track professorships, which is fantastic. I've just provided 2 examples at the bottom of 2 projects we have funded, one being the low-cost thermostabilization of a Rift Valley fever vaccine for veterinary use. And this has had real significant impact. This was a project using the ChAdOx vaccine vector platform. They were able to show that they could create a thermostable vaccine using this ChAdOx platform. And so although this project was focused on around Rift Valley fever, the impact of those outputs means that when people are creating target product profiles for the ChAdOx platform now, they can use this data to show that it can be thermostable at temperatures. So it's really, really wide reaching. And another key example is we funded a project on efficacy testing of novel emission and oral vaccines for Aeromonas hydrophyllae and tilapia and Vietnamese catfish. So we really tried to support the aquaculture field as well. So our African Schools Outreach Programme is a slightly different activity. Research has shown by the International Food Policy Research Institute that there's a really low representation of women in agricultural research in Africa. And so from this, IVVN was able to bid for some funding to support women researchers out in African countries to provide some school outreach work. And this has again been going for several years now. It's really, really run by the ladies out in the African countries of Kenya, Uganda, Cameroon, and Nigeria. And what they do is they take a lab in a suitcase, everything's in the suitcase, to these schools. They do tours of the schools, really teaching the children principles of veterinary vaccinology and laboratory skills. In the hope that showcasing, science to school children to get them interested. And of course, there's a particular emphasis on the fact that the people running this are all women. So it's showcasing that women work in veterinary vaccinology and work in science in general as well. So, so it's a great sort of, at the early stages, educating children on veterinary vaccinology. And I really urge you to have a look on LinkedIn, because they're very, they love showcasing all the work they're doing. They've made videos and presentations on it, and it's really, really fantastic what they've achieved. Okay, so yeah, finally, I wanted to just highlight again that we are a key knowledge hub for the community. As I said before, we run these newsletters once a month filled with information from the community. Again, there's a QR code there to register for the newsletters. We also run webinars which are geared up to showcase key stakeholders working in the veterinary vaccinology field. So last week we held one on the Centre for Veterinary Vaccine Innovation and Manufacturing that's based at the Pirbright Institute in the UK. And we try and run these once a month, just really showcasing what stakeholders do and how community can get involved. And of course, we're active on LinkedIn, so please do give us a follow. And I will just leave it again there. There's some QR codes for you to see in terms of our membership and our newsletters. And the IVVN email address there if you ever wanted to follow up with us. Thank you so much for your time, and I really hope you enjoy the conference over the next few days. Thank you. Bye.
Now let's welcome Marlene Brass from the International AIDS Society. Marlene is director of HIV programs and advocate at IS. As we know, IS and its vaccine program for global HIV prevention and vaccine research. They promote scientific collaboration and policy innovation to accelerate the development and implementation of HIV vaccine worldwide.
Good morning, good afternoon, good evening, everyone. I'm Marlene Bra, Director of HIV Programs and Advocacy at the International AIDS Society. Unfortunately, I cannot be there in person, but delighted to present what the IS is doing in the field. The International AIDS Society is an independent membership organization that unites HIV professionals around the world. At the IS, we believe that progress happens when science, policy, and activism come together. Through our major HIV conferences and programs, we unite researchers, clinicians, policymakers, and communities to share evidence, influence policy, and drive impact. One of the areas where this interaction of science, policy, and activism is most needed is HIV vaccine research. It's a field that brings together cutting-edge science, long-term vision and commitment, and deep community engagement. Despite remarkable progress, HIV remains a persistent global challenge. More than 4 decades into the epidemic, nearly 41 million people are living with HIV, and 1.3 million new acquisitions were reported in 2024. These numbers make very clear that, despite decades of innovation, the world is not on track to meet the UNAIDS target for ending AIDS as a public health threat by 2030. Antiretroviral therapy and pre-exposure prophylaxis have revolutionized HIV prevention and treatment, saving millions of lives. Yet, inequity in access, fragile health systems, and persistent social and structural barriers continue to limit their reach, especially in low and middle-income countries and among the most vulnerable communities. The current global funding crisis has amplified these vulnerabilities. Declining investments threaten the continuity of prevention programs and could reverse hard-won gains, as shown in this projection where HIV acquisitions rise sharply if funding is not restored. Vaccines in particular represent more than a scientific achievement. They are a cornerstone of health sovereignty. By empowering countries to protect their populations through local research, manufacturing, and delivery capacity, vaccines reduce dependency, strengthen preparedness, and promote long-term sustainability in public health responses. An HIV vaccine would not only be a breakthrough for epidemic control, but also a catalyst for greater equity and autonomy in public health. As Peter Piot, former executive director of UNAIDS, once said, we will not treat ourselves out of the AIDS epidemic. This statement captures a fundamental truth. While treatment has transformed HIV from a death sentence into a manageable condition, it has not stopped the epidemic. A comprehensive strategy is needed, and one that includes prevention, treatment, and ultimately an effective vaccine. Historically, vaccines have been the cornerstone of ending infectious disease epidemics, and HIV should be no exception. Vaccines are cost-effective and sustainable. They allow long-term savings, reduce treatment burden on our health systems. Vaccines are integrative and synergistic, and an HIV vaccine could work alongside ART, PrEP, and STI prevention. They are socially transformative. Vaccines allow to reduce stigma, empower communities, promote equity. A vaccine could reach populations who are currently underserved or hard to reach by treatment and prevention programs. And they're clinically powerful in preventing new infection, co-infection, and associated disease. Vaccines, and I'm sure you will all agree, are essential for epidemic control. They are the only realistic route for ending HIV transmission globally. A vaccine offers a path to epidemic control and eventually elimination, a goal treatment alone cannot achieve sustainably. It is the long-term, game-changing solution that can end the HIV and AIDS epidemic. To date, no vaccine candidates tested have induced broadly neutralizing antibody responses or effective CD8 T cells. The first modestly effective HIV vaccine clinical trials was the RV144 trial in Thailand, resulting in an approximately 30% efficacy. Though modest, it was the first signal that a preventive vaccine for HIV is scientifically possible. Later, the AMP studies provided another breakthrough in understanding. While they did not reduce the risk of HIV acquisition overall, it did show a reduced risk in HIV acquisition in a small subset of HIV strains of the virus that were sensitive to the infused antibody, confirming that neutralizing antibodies can, in principle, block infection. From the RV144 trial in Thailand, which, as I was mentioning, showed modest protection and gave us our first positive signal, to the most recent Mosaico and Imbokodo studies, which, while they did not demonstrate efficacy, did reshape our understanding of immune response and viral diversity. The journey toward an HIV vaccine has been scientifically rich and at times humbling. Today, the field is entering a new era with mRNA vaccine platforms, structure-based and AI-supported immunogen design, and broadly neutralizing antibodies guiding next-generation strategies. Each of these innovations and trials, whether they're successful or not, builds directly on decades of effort, an essential step toward the scientific insight that will make an HIV vaccine possible. So where does IS fit into this evolving landscape? IS serves as a neutral convener that links vaccine researchers, vendors, policymakers, and affected communities. We facilitate cross-disciplinary dialogue, integrate new technologies into HIV science discussions, and advocate for vaccine equity, ensuring that research outcomes translate into accessible solutions for all. Our conferences have become a critical hub where immunologists, virologists, social scientists, policymakers, and community representatives exchange ideas that shape the global agenda. IS advances HIV vaccine research through 4 key areas: bridging community and science. We translate complex research into accessible knowledge, foster dialogue between scientists and advocates, and ensure community perspectives help shape research priorities. Through literacy programs and advocacy for ethical engagement, we make HIV vaccine research people-centered, because scientific innovation must always be guided by the communities it serves. We promote collaboration across disciplines. We work closely with partners such as IAVI, HVTN, AVAC, WHO, and regional research networks to share learning across fields, from COVID-19 vaccine development to synergies between HIV vaccine and cure research. We support the next generation of leaders. We have HIV vaccine science and advocacy academies that allow to empower early to mid-career researchers and advocates from Central, Eastern, Southern, and Western Africa, equipping them to become independent scientists and dynamic change makers. Our mentorship program pairs early career advocates and researchers with seasoned experts, providing one-on-one guidance and opening doors, we hope, and nurturing careers. These initiatives foster South-to-South collaboration, equitable partnerships, and a pipeline of inclusive, globally representative talent. And we try to make sure that there is a global voice in the field. While the HIV response is undergoing a global shift, including changing donor priorities that could threaten long-term investment, the field is entering a rich era of scientific innovation. The IS ensures breakthroughs like mRNA platforms, improved adjuvants, and precision immunology are shared across sectors and geography. We believe it is more important than ever to advocate regionally and globally in Europe, Africa, and internationally for sustainable domestic financing and supportive policies, safeguarding HIV vaccine research for the long term. The photo on the right captures this in action. You can see advocates and scientists learning together in Zambia, embodying all four pillars: connecting science and community, fostering collaboration, supporting next generation leaders, and training participants to become informed voices at national, regional, and global levels, advocating for sustainable HIV vaccine research. Looking forward, the field faces both challenges and opportunities, and our collective mission will be defined by a set of shared priorities that will guide scientific progress, equitable delivery, transparency, and sustainable financing. Scientific innovation, advances in mRNA platforms, AI-driven antigen design, improved adjuvants, and precision immunology provide powerful tools to accelerate vaccine development. Innovation should remain at the heart of progress. Data transparency, open and timely sharing of data will accelerate collective learning, allowing the field to adapt quickly, identify promising candidates, and avoid repeating mistakes. Equitable delivery, the future of vaccines must reach everyone who needs them, and not just those who can afford them. Addressing disparities in access is essential to achieving global epidemic control. And finally, adequate and sustainable funding. Long-term investment is more critical than ever before. Sustained financial resources ensure that scientific advances can be translated into effective vaccines, that trials continue without interruption, and that innovation is installed by shifting priorities. VIS continues to advocate for policies, partnerships, and financing mechanisms that embed this principle at every stage, from discovery to delivery, ensuring the HIV vaccine remains vibrant, equitable, and sustainable. The pursuit of an HIV vaccine has indeed been long and complex, but it is also deeply inspiring. Every trial, every new scientific insight, and every partnership brings us closer to our goal. At the IS, we see this as a global effort. Not single organization, country, or researcher can achieve it alone. The momentum we have today, from technical innovation to empowered advocates and researchers, gives us reason to be optimistic. Ultimately, the message is clear. Possibility is within reach, and it will take all of us working together to turn it into reality. This is the moment to celebrate progress, reinforce commitment, and continue building the partnerships, innovation, and leadership that will make an HIV vaccine a reality. I'm going to close here, and I really hope to see all of you, all key stakeholders and partners at AIDS 2026 next year in Rio as we collectively rethink, rebuild, and rise. Thank you very much for your attention.
Next, welcome Institute Butantan from Brazil. With 123 years of history, Butantan works to prevent diseases like COVID-19, influenza, tetanus, diphtheria, hepatitis A and B, whooping cough, dengue, HPV, and rabies. Today, we are very happy to have their pre-recorded video introducing their work in different aspects of HIV vaccine and development.
Production, research, culture, education. Instituto Butantan is all that and much more. With over 120 years of history, we are well aware of the responsibility of taking care of people's health. Our antivenoms, antitoxins, and vaccines have saved millions of Brazilian lives, making us one of the largest producers of immunobiologicals in Latin America. We produce 100% of the flu vaccines distributed by the Brazilian Unified Health System through the National Immunization Program. We are responsible for most of the hyperimmune sera used against venomous animals, bacterial toxins, and the rabies virus. Our hospital, Vital Brasil, is referenced in the treatment of envenomations by snakes, spiders, scorpions, and other venomous animals. Our bioindustrial center is in constant expansion. We have labs, factories, and buildings for production, filling, labeling, and storage, as well as a farm where we collect plasma from horses for the production of antitoxins, antivenoms, and equine rabies immunoglobulin. Our industrial park is prepared to operate efficiently and compliant with good practices, meeting the requirements of regulatory bodies. In the beginning of the COVID-19 pandemic, in 2020, Butantan's vaccine was the first to reach the arms of the Brazilian population. With the support of the Pan American Health Organization, we export our vaccines to other countries, consolidating our international recognition and our commitment to public health. To expand our vaccine portfolio, we work with several international partners. We are one of the largest biomedical research centers in the world. Constantly looking for new drugs, we have ongoing clinical trials for vaccines against dengue, chikungunya, among others. We also have a new, modern, and innovative laboratory complex that combines cutting-edge research and scientific dissemination to support the conservation of Brazilian biodiversity. Considering the importance of research and education in the development of Brazilian science, in 2018, we inaugurated the School of Higher Education of Instituto Butantan, which offers extension courses and graduate programs. We are part of the São Paulo State Department of Health and are located in an extensive green area where we have our science park, a cultural complex with more than 20 attractions, including several listed historic buildings, a forest park, and the first vaccine museum in Latin America. Leisure and scientific education together in a park that is remodeled, accessible, and prepared to receive visitors of all ages. We are proud of our centennial memory. We keep evolving, eager for innovation. We make science and promote health because our legacy is your future. Instituto Butantan, always in the service of life.
We hope you enjoyed today's program for ISV Global Vaccine Partnership Program. We'd like to hear your feedback. We are looking forward to have more partners joining our program. See you next time.